So it's a pleasure to introduce the report back session from the breakouts, which I think were incredibly lively, and I really enjoyed the cellular decision making one that I was in. We're going to start off with the recording systems lineage tracing session, and that's going to be presented by Sam and Nozomu, and then we'll have a discussion afterwards. So over to you two. Thank you.
Okay, thanks very much. Yeah, incredibly lively discussion around lineage tracing this morning and the opportunities we have in that space and really the technical challenges that we need to overcome. So just to cover some of the main discussion points and questions here, we were covering the status of current and likely next advances in lineage tracing technologies, what we're learning about the best approaches and major challenges in mapping clonal structure and cell lineages during development specifically, which other modalities should we integrate with lineage tracing? Right now it's very transcriptome centric. How do we build from that? What opportunities exist for lineage tracing in vivo? So lineage tracing in vitro is quite accessible, but how do we translate this to in vivo developmental systems? The current methods are somewhat focused on clonal analysis. How do we turn this into hierarchical lineage trees? How do we think about continuous lineage tracing as well to increase resolution? How should lineage tracing experiments be designed? And this is something I'll cover shortly in some of the technical limitations. What artifacts should we be cautious of? Fundamentally, how do we apply these techniques to understand human development? So using natural somatic mutation or mitochondrial mutation tracing. And what can we learn about human development? So right now we're in this kind of technical phase of applying lineage tracing, but what is the biology that we're really going to learn from this? And how do we integrate other mammalian organisms into our understanding of human development? So as part of the biological questions here, so a lot of the discussion focused on the biology, which was fun. So I'm going to start with this. First of all, can we align mouse lineages to understand more about human lineages? Also, can we move to the blastoid or gastroloid systems that have been discussed in other human cell atlas meetings? Are these faithful models of human development? Is lineage tracing of these systems going to be useful? Also, we think we should engage more with the Evo-Devo community. What can we learn from lineage tracing, a wide array of different systems? And then how do we align these different lineage trees? So then this comes into, is development or is a biological process stochastic or deterministic? Is it biased? How do we lineage trace many different replicates and then combine that data computationally in a way that we can understand something deeper about the dynamics of that system? So we were discussing some of the limitations of this, that by kind of combining over different biological replicates, that may hide some of the interesting biology that's happening with these systems. One of the technical limitations there is dropout in these systems. How do we address that? So we'll come back to that shortly. Okay. Apoptosis. This is something that we can't currently pick up with our lineage tracing systems, but it teaches us a lot about the biology, the discoveries. So we did a little bit of soul searching and asked, well, what interesting biology have we learned from applying these systems so far? And that really came back to some classic developmental biology, which was a fun chat. So some of the classic C. elegans lineage tracing experiments are the observations of convergent and divergent evolution in cell identity. So we've been surprised that cells behave in unexpected ways, but this is something that has been reported before in developmental biology. But I think now with the lineage tracing, we have the opportunities to find why do these cells behave differently? How do they converge onto, potentially a different track than they were originally assigned to in development? And we have the technologies now to dissect some of these molecular mechanisms, and we think this will be informative for biology, but also perhaps for studying disease as well. How do we keep cells on the right track, and how do we prevent them from going off route in the first place? So really some fun biology discussion today. This led into some of the technical challenges that we need to overcome. So while there's a lot of promise in the lineage tracing field, the more refined these technologies become, the deeper we're digging into the potential artifacts that we could come across and how to deal with these. So one big topic of discussion here was, how do we make this technology more accessible to the community? And so we were discussing AdGene, protocols.io, GitHub. So how do we make the experimental reagents available? How do we distribute them in a reproducible way? But then also, how do we engage the community computationally? So there was a lot of healthy discussion around that. So technical challenges regarding mitochondrial, tracking mitochondrial mutations. Again, a lot of promise in that relative to somatic mutations because they're relatively sparse, but there's still a lot of potential data hurdles there. There's just a general issue of low sampling with these current technologies. So a lot of the lineage trees we're producing are relatively rudimentary. So how do we build these continuous lineage trees? And then what do we do with the data once we receive that? How do we benchmark the various lineage tracing systems? So we said if there was, we looked to other examples of communities built around new technologies. And DropSeq came into the discussion a few times. So that was an example where the technology was released and a community coalesced around that very, very fast. And then it just really turned into its own animal where people were helping each other. One of the challenges in the lineage tracing field right now is there's so many different technologies. How do we build a community around multiple technologies so we can learn from each other? So I thought that was a really good discussion to potentially build on. Strategies to increase resolution were discussed. Also, the current lineage tracing techniques, we completely lose spatial information. So that means we can't observe how cell-cell contact, cell signaling contribute to defining lineage, which is incredibly important. Also, we discussed the limitations of clonal resampling assays. So when we barcode cells and we recover cells at multiple time points through an experiment to build a lineage and also to reconstruct what we refer to as state-fate relationships. So in these experimental designs, we're relying on capturing clones from an early time point and then replacing surviving sisters. But with the cells that we capture, we're relying on them to faithfully read out the molecular state of the sister that's still in culture. And this makes the huge assumption that there's no asymmetric inheritance of lineage determinants. There's no transient heritable memories that can shape cell identity. So I think some of these experimental designs are fundamentally limited, and we would benefit a lot from continuous lineage tracing. So we were really at that point looking more to the kind of classic live imaging lineage tracing fields and what we may be able to learn from there and kind of integrate these high dimensional multimodal measurements that we can capture now. Again, another big technical limitation we discussed: how do lineage tracing methods potentially alter or perturb the biological system that we study? So we spoke about building the essential controls into these experiments so we know, or we can assess how the system is perturbed potentially. We also spoke about silencing of the barcodes that we introduced by lentivirus. This is a big limitation for some systems, such as introducing barcodes into pluripotent cells and differentiating them, or introducing them into early developmental stages, and then they get silenced. So then you don't have any lineage. Also, the limitations of systems that there are very few cell divisions. They are not easy to work with for lineage tracing. So we have these technical hurdles that will be system specific. And as a community, we think we need to come together and address how we create some rules of lineage tracing. Yeah, so that sums up the session, and I'd like to thank everybody who participated. And Nozomu, do you have anything to add?
No, you covered pretty well. Just a little add to the how we can map the human developmental lineage. I think the point of bringing up Evo and thinking about how we can align trees of different species is pretty important, because in human development, we can trace the lineage only from somatic mutations, and we cannot introduce DNA barcodes, whereas mouse, maybe we can do, introduction of barcodes, high resolution lineage reconstruction. We somehow need to kind of, consolidate this information, and where we need to think about kind of computational approaches and also kind of evolvable perspective, because people say that, the early developmental lineage across species are pretty similar, so we can probably learn from there. Yeah, I think that's it from us.
Wonderful. Thank you, Sam and Nozomu. That really appreciate it. And that's a fantastic summary. We now welcome questions from the participants, from the panel members here, and we have a time for discussions. I guess one thing that I'd be curious about is, Nozomu, you said that the early cell-fate decisions are probably similar between human and mouse. I mean, how, there are like rough similarities, in terms of germ layers and so on and so forth, but at the molecular level, there are also differences like, yeah, anyway, we can go into that at the end. I was thinking of, transposable elements and stuff like that. Sorry, I've just realized that we go on to the next session, which was the cell fates, and then we'll have a big questions at the end, once all the breakout session leaders have reported back. So thank you very much, both, really appreciate it. And now we'll hear from Anne and from Tomonori, who jumped in at last minute, so very much thank you for that. And over to you, Anne.
I would like to share my screen to report on the lively session we had. It was led by Tomonori and myself. Alev was not able to join for a medical emergency. And what we discussed was understanding cellular decision making during development. So it's two fields meeting each other. All the single cell studies and along traditional developmental biology inheritance, where there are many questions already and not all of them have been answered. The first thing we started by doing was trying to agree on what decision making is. It's moving one cell to another state. The cell has to decide to do so. And one state and another are not just two cell types. They can be intermediates, but they can be also behaviors of cells, like a cell decides to move, a cell decides to send a signal or something like that. So as a schematic here, you can see a cell like a progenitor. It has a certain potency. We have been developing tools in the community together to study potency. Very often epigenetic marks are used as a measure of potency, but there are also functional assays. Then the cells go through several differentiation steps, or several steps to a final state. We discussed whether these were real steps or whether it was us discretizing the system, and what is the speed at the different steps for which we have computational tools where we can start addressing this question as pseudo-time. And more work trying to link having time points in the same system, which are closer and closer to better describe the links between steps in the system. We also discussed, and I will get back to that, how to link gene expression as measured in single cell studies to cell behavior, the ability of a cell to secrete a signal, the ability of a cell to migrate, and many other descriptors of cell behavior. We discussed which period we should be probing, which period of life, whether we should probe early development, organogenesis, perinatal development, and we've decided not to prioritize any and feel we're a big enough community. So that's what's important, is to put our observations together. A traditional also question in the biology is, in this decision making, how much does the starting cell know and relies on its intrinsic controls that will be deployed, and how much does it rely on signals from the environment which are extrinsic? And to address this question, we need to have information about what are these extrinsic signals, what are the neighbors of the cells, how many of them there are, how far they are, and what are they secreting. There are more and more tools that allow to address these questions, not only in looking at two neighbors and the signals and the receptors they have, but now the abilities developing to be probing the pathways, the signaling pathways that are elicited in the cells during the decision making. We try to launch a discussion on the commitment process, how a cell is committed, whether it's returning, but it didn't go very, very far. Nevertheless, we think it's an important thing to consider. So all of this is pointing to the idea that what is important and definitely relevant to this community and development is that the system is a dynamical system. And if we want to study dynamic, we need to probe different time points and space, but also have systems where we can trace cells, linking to what Samantha reported, but also do live imaging. And link also to model systems where this live imaging can be done, or develop in vitro systems where it can be done. So how to probe this dynamic process? We agreed on the fact that we have in vitro systems based on stem cells, based on tissue explants or cell explants. And it's very important to do benchmarking as a measure of similarity and develop actually measures of similarity. So there are some which need to be disseminated in the community, and we should also develop them further. To what extent the system we have in vitro is similar to a system in vivo, and how can we go back and forth? We test something in vitro, and then we check whether in vivo it's relevant or not. Live imaging allows to follow single cell and to know one cell, what they do next. And it's a multi-parametric space of what the cell, there are features that we can be described. How can we relate them to the features we see here in single cell sequencing, for example? That's a great challenge for the future. In the environment, very specific to humans or vertebrates, we think that the environment is not just the other cells, but also the signals from the mother. And interactions between the fetus and the mother is something that would be very interesting to study further. It's challenging, there's limiting material. If you start with stem cell systems, you can get more material, but there's an engineering step which requires more benchmarking to validate the system. Another thing we found was interesting, very important, is to be able to do perturbation of the systems. We can do a lot of perturbations, knockout, more milder perturbations, we should pay attention to categorizing perturbation types. The responses are complex, and it was pointed out that delineating modules of behaviors, such as a collective migration, such as a type of response to a signal, would be important for the future. So now to finish, the last point of the discussion was what other fields do we need to involve and bring in? And we came up with 3 important fields. First, to bring in theorists from the physics, from computational biology, who can help us conceptualize development and simplify. In this endeavor, it's important to gather quantitative data and think of how to link the data, the molecular parameters, to what we are conceptualizing. The second field we would like to engage more and more is bioengineering. If you consider the environment of the cells, the matrix is something we should have more and more control over. We can also, with bioengineers, put constraints on the cell numbers we have in an engineered system in vitro to study small groups of cells and how they interact. Also control better the space and their arrangements. We know that the interface between 2 cells, for instance, is important to define the response they will have. The last field we think is important to bring in is synthetic biology, people who are more used to controlling systems. We didn't go into many different aspects of synthetic biology, but we discussed a lot of optogenetics. Optogenetics can allow to spatially control single events, cell behaviors. We also discussed the fact that the bioinformatics and the responses are complicated after these optogenetics manipulations. That's something that we're going to have to learn to deal with even greater complexity that we've been dealing with. So with this, I would like to end, maybe add, Tomonori, do you have anything that you would like to add that I may have forgotten?
No, everything is covered by Anne very nicely. And I want to point on one point. I was very impressed about someone's comment about we need to conceptualize and make a theory for cell decision making based on the data, quantitative data. So it's a very important point, I think. So I want to add this point. Thank you very much.
Yeah, and I would like to thank all the great people we had in discussion who really generously shared ideas. Great community.
Thank you both. Yeah, it was a really fantastic session. I can vouch for it because I was there, and thank you for that great summary. So it's very clear. We are going to move on to the third one, which is Arnold, Kriegstein, and Seidman, who've joined us now. And what we're going to hear from them is a summary of the breakout on charting development all the way through the lifespan to aging. And I'm excited to hear about that. Over to you, Arnold and Cricket.
I'll publicly apologize to Sarah, who I gave a little premature aging when I was in the wrong room, and apologies for that. But we had a very lively discussion, which really focused on the complexity and diversity of studying organs, and indeed all organs, from fetal development all the way through to the aging phenotype. And I think the key comments that we heard really began by recognizing that development is truly a continuum. And when we hear the very elegant studies about cell trajectories, we recognize that will provide incredible rich opportunities to understand this trajectory far more in far more detail. At present, there's a lot of focus on capturing particular key points: sometime in fetal development, newborn childhood, and then aging. But it was very clearly pointed out quite eloquently by many of our speakers that that really misses many, many important points that we should consider, not just for all organs, but certainly for specific organs. So for example, in the field of cardiology, the whole hemodynamic organization and structure of the heart changes from the day before birth until the day after birth. And to understand where those changes impact biology, we will have to capture each of those different points. In addition, we heard from the period of time in human development of about 1 to 3 months when there is a phase called mini puberty, in which FSH is highly expressed and shapes activation of the genital system and can lead to normal, precocious, or delayed puberty later on, and also impact aging. And then, of course, we recognize, we women at least recognize, there's the phase of menopause, which again clearly will have very profound impact on many organ systems. And so if we are going to fully understand trajectories, we're going to have to understand a lot of nuances in terms of the life of we human individuals. Chronological age, however, is not going to be a sufficient surrogate for us to really use as a demarcation of when different stages occur. It would be wonderful to have serum and hormonal markers. Perhaps bone age will be a more appropriate measure of some different stages, as well as physical traits, particular Tanner stages that will help define when puberty is occurring in adolescence. But as we all recognize, these are largely unavailable or else not accessible because of de-identification of samples. And so one of the really complicated issues is how are we going to appropriately time samples? And we again look to collaborations within the other bioinformatic strategies that are coming online to be able to address this, because cell trajectories and epigenetic states may be more informative than a hard number of the chronologic age of the donor. In this context, we also recognize the important diversities that are going to come from race, ethnicities, and the like. And understanding those as well as lifestyle exposures is going to be key to making sure that we are making appropriate and not illogical assumptions that we are all truly doing things the same. Important consideration in this regard is how many samples do we need to capture? Obviously, in my family, they say you can never have too much butterscotch pudding, and we can never have too many samples, but how many can we realistically attempt to capture? And we had a very thoughtful suggestion by one of our participants that perhaps some data generated by GTEx might help us to provide some rough estimates of numbers from which you might more fully capture diversity that is within a spectrum of quote unquote normalcy as compared to just randomly looking for 4, 5, or 10 samples. In addition, we recognize that when we talk about race and ethnicity, that also won't be truly sufficient to capture the full diversity. And if there was opportunities to have GWAS information on these samples, that would certainly be very, very important. When it comes to comparing these samples, we want to use them obviously for human health and also disease. And one of the issues that will become important to consider is how do you define an organ as being diseased, especially when they come from a donor that is often deceased or may have another illness, and we are capturing nearby cells that we presume are normal. And it was presumed, it was suggested that it would be more appropriate to not take and separate healthy versus normal, but to have mixtures of these tissues and analyses so that we are looking at the diversity and a spectrum of from health to disease rather than a dichotomous trait. I think that's going to be really, really important when we think about aging, because I don't know that we have any knowledge of what constitute a truly aged individual. The numbers of different common disorders that people have as they approach 50s, 60s, and beyond, and the medications that they're taking for those common disorders are going to have to be considered as part of health if we are going to make a viable and useful collection of cell information. The last thing I'll mention before turning it over to Arnold is that we recognize the complexity of acquiring tissues across the compendium of age, race, ethnicities, and exposures without an international effort. And in that regard, anything and everything HCA could do to help deal with not only putting individual collaborators together, but really mounting public awareness of the importance of donating tissues and helping to address local regulatory requirements and national limitations. If we have the opportunity to collect from around the world, but are restricted because of national constraints, that really is very, very problematic. And as well, whether there is an opportunity to collect fully from those donor tissues, inclusive of genome sequences and not just the transcriptome, that will add to our ability to obviously interpret the data and to parse likes the same in individuals and donors that are different as well. We would hope that this sharing would become far more easy with advent of nuclear sequencing in comparison to fresh tissue isolation. And so it really seems to be an opportune time to address this. And in that regard, we heard a little bit about the developmental GTEx program by the NIH that may be one way to be able to expand on tissues that are being collected by all of us. And then we had some other discussions when I'm going to turn it over to Arnold to tell us more about those.
Thank you, Cricket. That was terrific. I hope I'm unmuted now and that you can all hear me. Oh, good, terrific. Well, I think Cricket did a terrific job of summarizing our discussion. I have very little to add. We did touch on whether organoids could serve as surrogates for developmental processes, either in health or in disease outcomes. And, we emphasize that stem cell derived organoids, which are different than, for example, the gut organoids, which come from adult somatic tissues. These stem cell derived organoids, such as for the brain, really reflect fetal or neonatal stages of development. And so they're very difficult and may not actually serve as surrogates for more mature tissues. It was also mentioned that for studies of aging in particular, it might require greater cell diversity than we might normally see in just a single organoid-derived tissue. Aging itself, as was emphasized by Cricket, involves environmental exposures, lifestyle differences, chronic diseases. These are all very difficult to reproduce in organoids, even if they were of the appropriate, age interval. There's also inflammation, which was mentioned by one of our participants as an accompaniment of a variety of disease states. And that may need to be accommodated. Possibly it could be done by combining inflammatory cells with organoids, which people are beginning to do. And then the organoids often highlight cells that are really grown outside of their normal environments. And so that may limit the utility of organoid models for studying aging or critical epochs of developmental change. The other thing is that there's a great potential for functional studies using organoids, but they really require benchmarking or validation against primary tissue. And in that regard, a pediatric human cell atlas could really go a long way to helping inform organoid studies. And in fact, we think that's going to be essential for validating and benchmarking studies that are otherwise done entirely in organoids. Other than that, I think that, as I mentioned, Cricket touched on all the other topics that we discussed, and be happy that we maybe touch on other ones when we open up our discussion. The one thing I did want to mention, though, was I was very struck by the fact that we had global participation in our session. We had voices from Europe, the UK, and from South Africa, for example. And it became clear that the standards do vary across different regions around the world. And to give one example, when we collect elective terminated fetal material, the ages that procedure is done varies in Europe compared to the US. And it really emphasizes that a global partnership and collaboration might be required to really create the kind of human cell atlas that we're hoping to do for all the organs that we're studying. And so breaking down those barriers for sharing resources, tissue resources, could be really important. And I just want to emphasize again how the HCA could play a role in trying to make that come about. I think we're done.
Thank you, Arnold and Cricket. That's and those last points were really well taken. Great points, and hopefully the announcement from CZI this afternoon after this session will kind of get us more towards the in that direction. Okay, so we are opening up to the audience now, and invite everybody to chime in with questions, with comments, with discussion points. You know, Arnold's point about working together towards a pediatric cell atlas, you know, that's global, that covers all tissues, I think is a great one, and that's ambitious, you know, because of all the challenges. And certainly, you know, we need to synergize as a community to achieve that. So Gary has a question right here. Gary, do you want to, are you able to unmute and just ask, or should I read it out?
Sure. So I was just, you know, wondering about connections between these breakout sessions, and it struck me, Cricket, that you mentioned such a wide time range, and it really, you know, we really do have to think about a very wide time range of development in pediatrics. So from the technology side that we heard about from Sam and the Nozomu about lineage tracing, how, you know, I think that's being applied to kind of short time ranges. How do we think about that over a longer lifespan of humans?
Yeah, I think that we'll have to do that from obviously different donor samples that are treated as a single tissue, and look to see it, single cell map, and see if we can see the trajectory between as well as within an individual. And then another approach that was suggested in our group was whether that would be a very useful purpose for organoids, to see if we can age the organoid either by pulsing them with a stimulus or simply waiting in time to see if we can gain some insights in terms of this. But sequential sample, obviously, in the same individual is unlikely to really be possible in humans. I would flip it around in one other regard, which is that we heard earlier about using other organ systems, other organisms rather. And in that regard, I think we often go to the mouse, but this is where non-human primate might be an appropriate way to really have more consistent longitudinal data points.
I'd agree with that as well. In the lineage tracing session, we had the discussion of using non-human primates for some of these lineage tracing applications. So we're in agreement there.
There is, of course, the mosaicism, you know, that allows you to trace lineages in adult tissues. That's been very profitably applied to the brain, for instance. And, you know, I think that's a really exciting technology.
Sam, you discussed that, I guess, in your session. Is there anything you want to add? I mean, you mentioned that in your summary as well.
Yeah, so about using somatic mutations to trace lineage in human systems, about using mitochondrial mutations to gain higher resolution as well. And we briefly discussed a paper that came out in Nature on Wednesday, constructing phylogenies from humans using whole genome sequencing. So I think this is a really exciting area. And I think we can also supplement that with some of the organoid models and blastoid, gastroloid models as well.
Which double downs on why we need to have genomic sequence as well as these other sequences of our tissues.
Yeah, absolutely, I agree.
Yeah. Yeah. I mean, So Bruce is asking, Bruce, feel free to unmute. Bruce is asking how much daughter cell subtype generation is there in post-developmental stages in tissues that don't replicate highly. So, okay, so this is related to the somatic mutation lineage tracing. So
Yeah.
that I think that paper is quite broad. Yes, Sam, go ahead.
Yes, we discussed the limitation of this. So lineage tracing in systems where there isn't a lot of cell division, this is a huge limitation. You just can't gain much lineage from that, especially with some of the cell losses that we face, which I think speaks directly to that. So it could be easier in earlier development where we have lots of cell division, but in later stages, later in life, it could be difficult.
So that's very true of the heart and brain, in which at most maybe 5% of cells divide as one is getting into the second and third decade of life. And so it's really, yes, heart and brain, Bruce. And frankly, that's exactly why, even more so than the brain, Arnold, the heart doesn't get cancer. It just doesn't get cancer. And that's why we think.
So what it means is that your resolution is limited for those tissues, right? That you would get some information, but it just wouldn't be very finely resolved.
Right, except at the mitochondrial level, and mitochondrial aging does occur. Somatic mutations that we've looked for in human heart tissues are very, very rare, and we see them more perhaps in disease than we see in healthy. So there must be some somatic mutation, but it's not clonal in a way that is useful.
Mm-hmm. Anne, do you want to say something?
Yeah, I had a question on to Samantha and Nozomu. I think the lineage tracing gives very interesting information, but it would be very interesting to also compare it. It shows you what a cell does and its lineage, but it doesn't tell you anything about what it can do. And it links to a discussion we had in our session on organoids, for instance, are great systems where you can explore what the cells actually do, but also what they may never do in the body that they deploy when they are in these weird environments. Do you have any thoughts of that?
Oh, I have lots of thoughts about this. So we discussed at length this concept of constructing state-fate relationships through clonal resampling assays. So, you say you can retrieve a lineage, but you don't have an understanding of cell potential, or, how that cell potential is defined through molecular regulation from early stages, how that relates to the potential of that cell and what it will eventually become. So some of these lineage tracing experimental designs try to capture cells in their early stages, their sisters, replate the other sisters, and then they go on to do whatever they're going to do in development. But then you have that early capture of that early state and how it relates to lineage outcome. So in that respect, you can capture some of the information on cell potential, but it's limited to systems, so regenerative systems where we can tag cells, we can manipulate them, we can transplant them, we can recover them. But in organoid systems, it's incredibly challenging because we can't simply take cells out and put them back in. So take sisters away and leave sisters there. And so there's a lot of technical challenges around that. I don't know if that answers your question exactly.
Yeah, in part. One way is also maybe to link potency to all the epigenetic marks and associate your lineage tracing to the epigenetic state of cells at different steps. It would be interesting too, I think. And
I think this is why we need to map chromatin accessibility and transcriptome in parallel with lineage. So I think that's a huge missing piece right now. And there are many groups working on that. So it's an area to watch.
Yeah, another thought I would add here is that in CRISPR genome editing based lineage tracing, you are putting mutations to kind of DNA tape, right? And I think we need to boldly think about the potential of recording, like for example, RNA expression status to the same DNA tape. so that we can read out the previous status of transcriptome profile or something like that.
Yeah, I agree that it would be, the dream would be to have combinations of data kind of for the same, perhaps also methylation, which can give us some information on both cell identity or, cell state and then lineage as well, or other combinations. Any other questions? So Arnold, Gary, please, yeah.
So maybe just one last one. Arnold mentioned the need for a global effort to capture tissue from different stages, and I'm just wondering what other scientific advantages are there from sampling diversity across the globe? Like, there's a lot of environmental diversity, so what can we gain from having samples in different environments?
Well, I could maybe begin just by saying, in our group, we did discuss, what would constitute a normal cell atlas. I know there's an effort to create such an entity, and normal is very difficult to define. And the idea of incorporating samples globally would be a step in that direction, because there are environmental effects that we've mentioned, dietary effects. There are a whole range of different reasons why to try to get a normotypic cell atlas of any organ in human development might require integrating data sets from all these diverse populations, and the more diverse the better. And there are some barriers to sharing tissue samples that were mentioned in our session, including regulatory hurdles, which are going to be challenging to overcome. But I think it's an effort well worth trying, because we really need to develop some understanding of what it's like to be human, and what those boundaries are. And we need to have diversity in order to do that.
And if I could just add, we think about exposures, but food intake, just quantity of food, types of food, and frankly, stress of life. Imagine being in Kabul right now. These have got to have profound impacts on our biology, and I don't think we have a clue.
Great. Okay, I think that's a fantastic note to end on. And segging into the pediatric cell atlas. So I think we have a 15-minute break now. And then we will reconvene for an exciting announcement from CZI. Thank you so much for all the breakout coordinators, for all the breakout participants, and for the organizers here for a fantastic session. And see you in a few minutes. Bye.
Bye-bye.